everyone who has joined us today. It's lovely to see you all here. And as Sonalika very accurately mentioned, that we are going to indulge more into this wonderful topic of astrobiology or space omics. And I'm pretty much sure that every one of you who is present here today is interested and enthusiastic, and you're well aware of the various kinds of research going on around the world. There are also numerous opportunities which are early career as well as uh, for the more experienced ones, be it a postdoctoral fellow or a scientist position available for various people all across the globe to grab this opportunity. To help people do that and announce yourself in the domain of astrobiology, we at Fine Biotech have organized this particular opportunity for all of you, where every one of you can get an opportunity to indulge in your own independent as well as team projects, where you would also receive mentor support, one-on-one guidance, and available course materials, which would help you to learn all along the way as you go. And if you have joined us for the first time, there was another webinar which was done. And in that webinar, me and my colleagues have discussed in detail about what are the topics that we are going to cover, what is astrobiology all about, what is our focus, and what is our aims. And in today's webinar, which is more of like a discussion, we would be discussing about all the project ideas that you guys have and what are your aspirations. We would also discuss about potential teams. You guys can form teams amongst yourselves, those who are already here, or if you have a research lab or a team who would be interested, you can also invite them. And today we will be discussing about where to find the data sets, what the data looks like, and how can you start your own project just by sitting at your home. This is a pandemic hit era where it's very difficult to get hold of a laboratory, to go to the fields and do the research work. Same kind of problem everyone is facing all around the world, but we can't stop, right? We have to go on doing our own projects and coming up with a publication, participating in conferences. And therefore, this sort of a model where you can actually get involved in practical works right from your home remotely provides you an exciting opportunity to be a part of bigger consortiums and groups just by sitting at your home. So before I move on to the further discussions, I would like to show you guys about what the entire plan is, how it looks like, and what is the plan altogether. So I guess you guys can see my screen. So this is the homepage of the program, which is called Introduction to Space Omics. And this program is actually designed you to help you through your research project. Now, there are several steps in defining a project and getting it done. For example, it begins with finding a good data set, a data set which is worthy of looking deeper into, to curate it, to label it properly, and to make it ready. The second part is obviously the analysis part, the methodologies that you have to use, and how you're going to use it, what is the appropriate methods that you should apply for your expected results. Now, once you get the hold of the results, how do you understand if the results are a good result or a bad one? Or what do you biologically interpret from those data? And how can it correlate with whatever is happening or going on in the biological body? So these are the various challenges which many students face in today's world. Whatever background you belong and whatever your expertise might be, these are the problems or the challenges which most of the people face. And that's why we have designed this particular curriculum to help you understand what we are going to talk about and what is it all about. So to begin with, the first day would be focused towards the topic of astrobiology. What is it all about? What are the various subtypes, as you can see on my screen? We will be also discussing about the theories of origin of life and the various topics or debates currently going on in this particular domain. The next day, which is on October 25th, we would be diving deeper into the concept of origin of life, because I'm sure that many of you are interested in actually analyzing or understanding the origin of life better, and you want to start some projects of your own use in this particular aspect of origin of life. So we would give you some idea about how you can gather data, what sort of data should you look into, what are the concepts that you can analyze. Definitely we would be talking about extremophiles, because you all must agree that extremophiles are very, very important in the understanding of origin of life or not, as well as in the concept of hoping for life beyond Earth. And we would just not give you another lecture, but also talk about the data sets. How does it look like, where you can find them, and also give you certain project examples which uses ancient RNA reconstruction or protein remodeling, and what results have they got, and what can you guys do as a part of your project. And this is not something which is very rare. Like, many research groups are actually doing marvelous research in this particular domain. Then the third day would be focused more towards the other aspect of astrobiology, which is space flight travel. So as you would agree that in the current era, with the commercial spaceflight agencies coming to the forefront, traveling to space has increased manifold. And with the increase in the number of missions to space, the risks of astronauts getting affected by the various factors of spaceflight is also increasing rapidly. For example, spaceflight is made up of various factors like microgravity, ionizing radiation, and isolation that has an effect on their brain. So before we plan a mission to Mars or maybe even the moon, which are long-term missions, it becomes mandatory to completely understand the effects of these conditions on the genome and the expression of genes, the gut microbiome of the astronauts. And we need to make sure that they won't suffer from any chronic illness like cancer when they are on that particular mission. To do that, it's the right time to actually get hold of these sort of data sets and start analyzing them. And believe me or not, there are various organizations in the world who are looking into this particular domain, and they want people to get involved in this sort of projects. To give you an example, there's one conference which is coming up next month, and it's focused towards space health and the peaceful use of space technologies. So you can understand by the title itself that people are all around the world are actually talking about this particular thing. So it's a best opportunity for us to get involved in these sorts of conferences and other opportunities by having a project of our own. So that's what we will be talking about. And on the fourth day, since Fine Biotech focuses on bioinformatics, how to utilize the various omics data to your project, we would be giving you a thorough explanation of all the omics levels, like genomics, transcriptomics, metagenomics, epigenomics, and also multi-omics integration. This omics levels were studied in the NASA twin study, as mentioned by Sanalika. But here's the benefit of doing it with us. First of all, you don't need to have any coding skills. Many students who are biologists like me have a problem with coding. That's a big hurdle, but here you don't need to code. Why? Because here you would get all the algorithms just in a point and click interface. So you don't need to know about any of these algorithms, what's behind that, what are the codes. You just have to see this particular user interface, which prompts you, helps you with every detail which pops up on the screen. And you just have to click, point and click, and you can build the entire pipeline. I would be giving you other examples as well and show you how to do it. And excuse me. So the final day, that is the day 5, would be again focused towards projects, because that's the day when you guys need to come up or decide on your projects and finalize what are you going to work on from this particular day. So this day I would be giving you other examples of projects and walking you through my project, how I do it, how I did it, and what my project looks like. Even today I would be showing you some examples, and on this day we would discuss with you all your confusions or doubts. And from this particular day, you would get the opportunity to work on your own project, and you would be meeting the mentors twice a month. So you would be getting the support. You can show them all your results, consult with them, and that final target, of course, is to come up with a good result which you can publish if it is excellent. If not publishing just now, you can definitely take it to a conference as a poster or an oral presentation. So let me give you an example of the various projects that has been already done by the people who have participated in the previous programs. So as you can see here, this is my project here. And if I scroll down, you would see that various other people have submitted their projects, which they have completed with fine. So you can see these research questions, and they are the ones which are going around in the world of biotech right now. So here you can see the in silico analysis of affected biological pathways in multiple sclerosis, transcriptomic analysis of CD4 plus T cells. So every project that has been done here has been either published in a peer-reviewed journal or has been taken to a conference, and several students have won several prizes, in fact. So let me walk you through one of the projects and show you how it looks like. So the project here, if you go through, you would find that it's a well-structured interface where you would find a brief overview. You would find details about the datasets. You can check it out. And these are all submitted to the Harvard repository. You can visit them and analyze them yourself. Then comes the abstract part, where you would explain your project in brief. As you guys know, the structure of a peer-reviewed journal paper, basically, it's similar. So once you submit your project in this particular manner over here, it gets very easy to take it to a publication or to take it to any conference, because everything that you need is ready. So it looks really interesting, right? So before we move on to you guys and discuss with you what are your interests, and I would also show you some techniques to analyze it and some pipelines that I have created. But before moving on to that, we have with us Dilara Dekan from Turkey. And she has been one of those participants about whom I was talking about. And she has already done a project in this particular domain of astrobiology, where she has analyzed the radiation threats and the effects of radiation, ionizing radiation, on the gut microbiome of astronauts. And she has also taken her project to a preprint publication, and I'm sure she is looking for submitting it into a peer-reviewed journal. So it's the best opportunity to have a look and learn from her, have a look at her project and learn from her how was her experience of the project, how did she utilize the platform of TBINFO InPine, and what's her entire experience, because it's a similar kind of experience that you guys are also going to experience in just a few days. So hi Dilara, over to you now.
Hello, thank you for that introduction.